Hello everyone, welcome back. In this video we'll be doing about the search for genetic material, which is the next subtopic from the chapter, that is molecular basis of inheritance. At that time, the search for genetic material began, and it was at first it was given transforming principle. Okay, transforming principle was given by the scientist called Frederick Griffith. Now how does he give this transforming principle? Is he was doing some experiment with a bacteria. Okay, and that bacteria is streptococcus pneumoniae. So this was the name of bacteria. He was doing some experiment with this bacteria. Some of the streptococcus pneumoniae, okay, he cultured this bacteria in a medium. Some of the streptococcus pneumoniae, they produces smooth colonies. Okay, smooth colonies which appear shiny, okay, and very smooth colonies, like that it goes on. Okay, and some of the streptococcus pneumoniae, they were rough. They did not produce any kind of smooth colonies. Now these smooth colonies are nothing because some of the streptococcus pneumoniae, okay, they produces polysaccharide coat. Okay, polysaccharide coat which appears very fine and smooth. Okay, which gives that appearance of smooth and shine. They produces polysaccharide coat. As a result, the some of the streptococcus pneumoniae, okay, suppose this is 1, this is 2, some of the streptococcus pneumoniae, they produces a smooth shiny colony, and some of the streptococcus pneumoniae, they do not produce any smooth colony because they do not have polysaccharide coat. They do not secrete this polysaccharide mucus coat. Okay, so this appear rough in texture. Okay, so this was named as R strain bacteria. Okay, R strain bacteria. Now R strain is streptococcus pneumoniae, and this was S strain bacteria. Okay, S strain bacteria, that is streptococcus pneumoniae. Okay, now what does he do is he did some experiment with this streptococcus pneumoniae. Okay, this R strain bacteria was virulent. This was virulent, and it was not non-virulent. Okay, it was not virulent. Virulent means nothing but the bacteria, okay, that can harm some other organism. When this bacteria is injected to some other organism, this bacteria can kill that organism by the disease pneumonia, because streptococcus pneumoniae is a bacteria, okay, that causes disease called pneumonia. So this virulent S strain bacteria, okay, they cause other organism pneumonia, and with that pneumonia the other organism can be killed. Okay, but this R strain bacteria, R strain streptococcus pneumoniae, they are non-virulent. That means even if they are injected to some other organism, even if they infect some other organisms, it cannot lead to death of that organism. Okay, so R strain bacteria was virulent and R strain bacteria was non-virulent. Now what does he do is he injected S strain bacteria. He is injected S strain bacteria to mice. Okay, injected to mice. Now injected to mice, and then he injected R strain bacteria again to another mice. Okay, now when he injected mice with S strain bacteria, mice, it was killed. Okay, mice died. Why? Because R strain bacteria were virulent and S strain bacteria were virulent. Now when another mice was injected by R strain bacteria, the mice was alive. Mice was alive. Okay, so again, now what does he do is he heat killed S strain bacteria. So what does he do is he just killed this bacteria, not mice now. Okay, at first he performed that, he injected mice with S strain bacteria, mice died. He again injected some other mice with R strain bacteria, mice was alive. Now he killed S strain bacteria. How he killed S strain bacteria is by heating them. Okay, he heated that S strain bacteria and S strain bacteria got killed. Okay, now then he injected S strain bacteria which was heat killed. That means the dead bacteria. Okay, the bacteria which was already dead because it was heat killed. Okay, the dead S strain bacteria, he injected to mice. Now mice was alive. Why? Because the S strain bacteria was not live, it was dead. So it was dead, that's why it was not virulent anymore. So when the dead S strain bacteria was injected again to mice, mice was alive. Now at last what does he do? Again he S strain bacteria which was heat killed, okay, the dead S strain bacteria plus this one R strain bacteria which was not virulent, the mixture of these two bacteria, okay, he injected to mice. Now what happened to that mice? If we see all these things, then we can conclude that mice must have been alive, isn't it? Because the heat killed S strain was already dead because it was heat killed, and R strain bacteria, it was non-virulent. But to his surprise, okay, the mice was dead. Mice died. Okay, he was surprised. Mice died. Now how this mice died? Okay, he at last then he concluded that something must have been transformed, okay, something must have been transferred from this S heat killed bacteria to the R strain bacteria, which make this R strain bacteria virulent, and then when it was injected to mice, it died. Okay, this Griffith, he concluded that something must have been transferred from this dead heat killed S strain bacteria into the cell of R strain bacteria, which made this R strain bacteria virulent, so that the mice was dead, mice died, okay, when it was injected. So this R strain bacteria must have been transformed, okay. He concluded that from S strain something must have been transferred. Transferred means from one place to another, okay, from the cell of S heat killed bacteria to R strain bacteria, and this transferred made this R strain bacteria transformed into virulent, okay. And when they are injected to mice, mice died. At first it was not virulent, but from the cell of this heat killed S strain bacteria, something has transferred into the cell of R strain non-virulent bacteria, which transformed this R strain bacteria and made this R strain bacteria virulent. Then mice died. So he concluded that. Okay, and so now see, he concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Okay, so some transforming principle is applied here. Okay, some transforming principle has been applied here. He concluded that. Okay, transferred from the heat killed S strain had enabled the R strain to synthesize polysaccharide coat. Something which was transforming principle that was transferred to R strain bacteria made this R strain bacteria to synthesize this polysaccharide coat and turned into virulent form. Okay, so this must be due to the transfer of genetic material. Okay, this must be due to the transfer of genetic material from this S heat killed bacteria to R strain bacteria, but the nature of genetic material was not defined from this experiment. Okay, how is that genetic material, what is the nature of that genetic material that was transferred from heat killed bacteria to R strain? It was not identified from this experiment. He just concluded that there must have been some transforming principle that transformed R strain bacteria into virulent. Okay, so this is how Griffith in his experiment proved that something has been transferred. Okay, thank you.